Structure of bacteria. Structure of bacteria comprise of some essential components. Essential components are those components which are present in all bacteria. And then there are some non-essential components. Non-essential components are those components which are not present in all bacteria. So the first essential component is cell wall. It gives the rigid support to the bacteria. This is the essential component. Then just outside the cell wall, we have a non-essential component, which is capsule. Capsule is not present in all bacteria, but capsule when present give pathogenicity to that bacteria. It makes that bacteria more pathogenic. Then an essential component just inside the cell wall is cell membrane. So the cell membrane basically has a role in transport of the substances just inside and outside bacteria. And then we have cytoplasm just inside the bacteria, and it has essential components like nucleoid, which comprise of DNA having role in cell division. And another important essential component is ribosome. So nucleoid having DNA and ribosomes. Here are ribosomes. They have a role in protein synthesis and they are essential component present in all bacteria. Then we have some non-essential component like plasmids. These are extra chromosomal DNA and granules. These are inclusion bodies which act as a site of nutrients. And then we have other non-essential components like pili. These pili are having role in motility, attachment of bacteria to the host surfaces, which makes them pathogenic, and it also has a role in motility. And flagella, which has a primary role in motility, is a non-essential component. Both pili and flagella are non-essential components. So as a whole, the essential components of bacteria comprise of cell wall. Just inside cell wall we have cell membrane, another essential component, nucleoid having DNA, and ribosomes. These are essential components, whereas the non-essential components are additional structure which may be present in some bacteria are capsule, and inside cytoplasm like plasmid, granules, and then pili, which are present outside the cell wall, and flagella are also present outside the cell wall. These are non-essential components which are present in some bacteria, giving additional features to those bacteria. Essential components of bacterial structure. Cell wall is essential component. Cell membrane is another essential component. Then we have nucleoid, which comprise of DNA, ribosomes, mesosome, and finally periplasmic space. Cell wall is made up of peptidoglycan and it has important role in giving rigid support to the bacteria. It is also the site of action of some antibiotics, for example penicillin, act at the level of cell wall. Then we have cell membrane. It is made up of lipoprotein bilayer, but it does not have sterol, that is, it does not contain cholesterol. Remember, eukaryotes, for example humans, have lipoprotein bilayer cell membrane, but we do have cholesterol, whereas bacteria lack those cholesterol. And cell membrane has an important role in transport of substances. Then we have nucleoid. It comprises of DNA and it has important role in cell division. Then we have ribosome. Ribosome is made up of RNA and protein in 50 sedimentation and 30 sedimentation subunits, and it has important role in protein synthesis. Then we have mesosome. It is basically formed by invagination of the plasma membrane, and it has important role in cell secretion and division. Finally, we have periplasm. This is basically a space just outside the plasma membrane in bacteria. And then we can see here how mesosome is basically formed. So mesosome, so you can see this is cell membrane, and this is then invagination of the cell membrane, and this is how invagination of the cell membrane formed the mesosomes. It has important role in cell division and secretion. Finally, we can discuss about the periplasmic space. So see, this is the cell membrane and the outside layer is peptidoglycan just outside cell membrane, and below the peptidoglycan cell wall layer we have this typical periplasmic space. It is also an essential component of bacteria. Now coming to non-essential components of bacteria. Capsule, for example, it is made up of polysaccharides. Capsule has many important functions, the most important being its role in giving pathogenicity, and it does this by protection against phagocytosis. It protects the bacteria from phagocytosis. And secondly, it also has a role in identification. How? Antibodies which are specific for capsule can help in identification of certain bacterial species. Then the antigens of capsule can also be used in vaccines. Other non-essential components of bacteria include Pili, Fimbriae, and Flagella. These three are basically closely related to each other. That's why I will discuss them together. See, this is Pili. These are hair-like structures. And then these are Fimbriae. These are smaller as compared to Pili. And then we have Flagella. These are the longest structures. Flagella are bigger than Pili, and Pili are bigger than Fimbriae. So you have to know that Pili are larger in size than Fimbriae. Fimbriae are the shortest, we can say. They are bristle-like. Pili are thin hair-like. And then we have Flagella. Flagella are larger than Pili, and they have a role in motility. Pili and Fimbriae both have a role in motility. They also have a role in attachment of bacteria to host surface, and they also have important role in conjugation, which is a mean of reproduction in some bacteria. Then finally we have few other non-essential components of bacteria, which include granules. These are also known as inclusion bodies, and they have glycogen, lipids, polyphosphates. So that means that granules or inclusion bodies are the site of storage of nutrients in the bacteria. Then we have plasmids, as we discussed. These are extrachromosomal DNA, and they encode various antibiotic resistance genes, and this gives the bacteria antibiotic resistance property. And they also have another role. Plasmids have been involved in antibiotic resistance and also in the production of toxins. And then we have spores. Spores have a thick keratin-like coat, and these are formed inside the bacteria only in cases of very disastrous condition, like in cases of very high temperature, very high heat. These spores are formed inside the bacteria, and they contain nucleoid and an important essential component, D-picolinic acid. D-picolinic acid is a calcium ion chelator. This basically gives the spore its resistance property against the heat. Very high heat and temperature, these spores are resistant because of this D-picolinic acid, and they have small amount of cytoplasm and nucleoid. And when the condition is favorable, proper temperature comes and there is proper water and nutrient supply, then these spores can germinate into a potentially pathogenic bacteria.